Hello boys, welcome back to another session of video tutorials. This session is meant for 11th August 2020 and this is meant for class 12th biology boys. And as usual, all of you know that our memory helps us in reflecting upon what we have been studying. In our previous session, we were discussing about the genetic disorders, in that particularly Mendelian disorders, and we understood how the Mendelian disorder called hemophilia, X-linked recessive inheritance, would cause lot of damage for us, resulting in bleeding. In this session, we are going to study about another important disease. This is called sickle cell anemia. Let me tell you, sickle in Kannada you say krupi, sickle cell. The RBC, as you are seeing in the photo here, red blood cell, will lose its shape. See, you can see the shape, normal RBC. Normal RBC, which is having this shape, will develop into krupi shape, sickle shape. So, the reason is, you can see there is a mutation, gene mutation is taking place. Because of that error in the gene, resulting in replacement of a glutamic acid. Glutamic acid is an amino acid. So, in place of glutamic acid, another amino acid called valine has come. As a result, the sequence of amino acids, see, in a normal person, this is the sequence. Whereas in abnormal person, look at the sequence, there is a change of sequence here. So in place of glutamic acid, valine has come, and that results in disshaping of RBC. So that's why we are saying sickle cell anemia. The moment RBC loses its shape, what happens is, inside the RBC, there is a pigment, red colored pigment, hemoglobin, HB, normally we abbreviate. So this hemoglobin inside has got iron. So iron combines with oxygen that we are taking, becomes iron oxide. So absorption of oxygen is because of hemoglobin, and hemoglobin is present in the RBC. Disshaping of RBC results in lack of oxygen absorption. So when oxygen absorptions are less, naturally our metabolism comes down. When metabolism comes down, we become weak. And whenever we become weak, that condition is called anemia, sickle cell anemia. People are becoming anemic because of RBC getting into sickle cell shape. This is what you should understand. So this sickle cell anemia is because of replacement of glutamic acid with valine. And what you are seeing here, all these, these are all nitrogenous bases in the DNA sequence. C for cytosine, A for adenine, G for guanine, T for thymine, the sequence, triplet code. So this triplet code results in the formation of amino acids, and the amino acids will have a sequence, and amino acids together will form a protein, and proteins will decide the characters of an individual. So the change in amino acid, you can see here, the change in amino acid sequence causes hemoglobin molecules to crystallize, becoming crystals. When oxygen levels in the blood are low, as a result, red blood cells sickle and get stuck in small blood vessels. So there will be problem in both the flow of blood as well as absorption of oxygen. So basically it is autosome linked recessive trait. Specifically, it is chromosome 11. As all of you know, we have got how many chromosomes? 46 chromosomes. Out of the 46 or 23 pairs, 44 are autosomes. Autosomes means bodily chromosomes. Chromosomes that will decide body characters. And 2 allosomes or sex chromosomes. Sex chromosomes or allosomes will decide sex of the individual. So out of the 44 autosomes, it is the 11th chromosome. Remember, you must remember what is happening. The sickle cell anemia is because of 11th chromosome. So this 11th chromosome sequence of amino acids, as you have seen, will be missing. So you must remember this for your UPSC exam, NEET exam, etc. So it is the 11th chromosome that will make the change. And I think we have come to another page. Don't worry. So all this we have covered. All of you know it, and we have completed this. It's a sort of review for you. Whatever I have explained to you, codominance I explained to you, IAIB blood group I explained to you. Okay, and you are able to see the inheritance of 2 gene. Okay, and then dihybrid cross, and you are able to see this. Okay, test cross, and then we have come to the next one, that is linkage and recombination, of course mutations, and yesterday's class hemoglobin, and then we were talking about the hemophilia condition. I explained to you how that the presence of gene H will make it, and here comes what we have been talking, right? And here I would like to tell you that it is the 11th chromosome, and it is controlled by single pair of alleles. Allele or pair of genes, HBA, HBS. Here A for absent, S for sickle cell. You must remember like that. So homozygous individuals of HBS, that is, if both the alleles are having gene for S, sickle cell, HBS, and if it is homozygous, then unfortunately the person becomes sickle cell anemic. But on the other hand, if it is heterozygous, okay, heterozygous means hetero means different. Different alleles are there, HBA, HBS. Then this person is unaffected as such, but then he has got potential danger. This fellow has got a carrier. He carries this gene, yes, okay. And defect is due to substitution of, just now we saw, the glutamic acid by valine. So this fellow is causing trouble at the 6th position of beta globulin chain of hemoglobin and 6th position of 11th chromosome, that you must remember. Due to substitution of single base, as you have seen, at the 6th codon, triplet code, beta globulin gene from CAG to GUG. So this is the change, A and U. So mutant hemoglobin polymerization under low oxygen tension causing change in shape of RBC. And normally you have seen biconcave RBC shape. From biconcave it become Kurupi, sickle cell shape. And what about the pedigree analysis? How do you know that a person is going to become sickle cell anemic? Now we'll take the center fruit of it. Now both mother and father, here we are taking it, sickle cell. That means this is, you can write it HBS and HBS, okay, because both are there. And so as this, mother also unfortunately HBS. Now you know, this is a bad luck family, I can say. So this allele gene and this gene combines HBS, sick person, sickle cell. And this combines with this combination, again second child also. And you take this and this, third person also. This combination of alleles results in this. So very, very bad luck family, unlucky family. So only the caution is that when the parents are having the genetic diseases, and unfortunately if both mother and father are having genetic diseases, the entire family suffers. Look at this, entire family suffers. So only whenever people are getting married, at least in European countries, they look for this. Whereas in India, we all know that we see the horoscope, we see the birth star and some calculations by Purohit, okay? But we need to, I'm not criticizing that, but people have their own faiths. But as a biology teacher, I would like to follow this. You must all know the genetic. And what is the best way to know the genetic disorders in a family? Pedigree analysis. Now let us take this case. Father has got a trait. That means father, I can write, he has got one gene which is dangerous fellow. He is there inside. And one is normal, HbA. Okay. And what about mother? Mother is good lady here. See, look at that. HbA, because she doesn't have the gene for sickle cell anemia. So she is lucky. Now the combination of this HbS and HbA, what happens? Look at that. Carrier. And HbS and HbA, again carrier, because sickle cell is there. And what about this combination? HbA from father side and from mother side HbA, normal. HbA from father, HbA from mother, normal. So the family is at least 50% lucky. 50% children are having sickle cell anemia, 50% are happy, safe. And we are also having both the father and mothers are having. We will take another case, this case, okay. Now father has got a trait, that means HbA and has to be HbS, correct? And so has this, mother also, HbA and HbS. And combination, look at this. This HbA, let's say, combines with this HbA. And the person is very, very lucky. Both are A absent. So normal child. Lucky fellow. Okay. And where are we? I think we missed. Yeah. And we are going back. Okay. Let me come back. Yeah. We are back. So here, as we were discussing, so the other combination, let's see. What is that? HbA and HbA we are seeing. HbA and HbS. So HbS, one has come, so carrier. Now this HbS and HbA, again carrier. And this HbS and HbS, very, very bad luck. Oh my God. Oh my God. Why is this happening? Okay, HbS and HbS. Both the chromosomes are having the S for sickle cells. This glutamic acid valence, no? So you can see here, the third one is the affected one, this one. So you can also try and see other combinations and see why it is happening. Like in the third case, the father is having sickle cell anemia. You know it is SBS, HB hemoglobin with the sickle, HBS and HBS. And here mother is normal, HbA, HbA. See the workout, this all are carriers. And another example we are seeing here, father with sickle cell anemia means we must be homozygous, HbS, HbS. Because unless it is two genes, two alleles, both are present, then only it becomes sick. And the mother is a trait, HbS and HbA. And you can see among the children, 50%. So 50% sickle cell anemia, 50% are carriers. Okay, that is the reason why